Good afternoon, and welcome to today's Museum of Science live program. My name is Eric, my pronouns are he, him, and I'm a member of our current science and technology team. And every year, we make our picks for the 10 most exciting, influential, important, and coolest stories from the world of science for the last 12 months. This year, 2020, has been very different in so many ways, but we still wanted to put our list together and share it with you, and we've been doing that all January long. We kicked off our countdown in early January with an honorable mention story. Didn't quite make the countdown, but our team was delighted to learn that scientists discovered the duck-billed platypus glows in the dark. Our number 10 story was the Atlantic hurricane season of 2020, which broke many records. Number 9 was the recreation of the voice of a 3,000-year-old Egyptian mummy. We continued our countdown with number 8, an interesting discovery that hinted at life on Venus. Number 7 involved the first ever dinosaur to be confirmed to be an avid swimmer, the Spinosaurus. Stay tuned on our social media for a related story with Massachusetts getting its state dinosaur. We finished with number 6 that week, awarding the Nobel Prize in Chemistry to 2 women for their groundbreaking work on editing DNA. Last week, we talked about our number 5 story, murder hornets, our number 4 story, the devastating wildfire seasons in Australia and the US, and our number 3 story, the triumph of astronauts launching to space from American soil for the first time in nearly a decade. If you missed those live streams, but you're interested in more details, you can catch the replays anytime on our Facebook or YouTube page. In a moment, I'll be joined by 3 of my fellow Museum of Science educators, and we'll finish our countdown with our picks for number 2 and our number 1 story of the year. I'll be your moderator during this session, which means I'll be fielding all of the questions that you ask our panelists. Please do begin asking questions about our stories or the process of how we selected them at any time. If you're joining us on Zoom, you can press the Q&A button at the bottom of your screen and include your name and age if you'd like a shout out to ask a question. If you'd like to show captions on Zoom, you can click on the closed caption button and select show captions. And if you're joining us on Facebook or YouTube, you won't be able to see your questions or comments, but we're happy you're tuning in. So at this point, I'll invite my panelists to turn on their cameras, introduce themselves, talk about how much they love the top 10, and we'll be ready to get started.
Great, thanks, Eric. Hi, everyone, I'm Megan. And I think what I love most about the top 10 every year is the chance to kind of look at all of these stories that come out over the year and just how much scientists are able to discover and figure out over just the course of a year. It's kind of mind-blowing. And that's what I really love about this process.
Hi, my name is Sarah. My pronouns are she and her. And yeah, I'm a top 10 junkie. I love all of the stuff that we've done. Probably my favorite just random story was last year's honorable mention was about rats being taught to drive these tiny cars. And I still go back and watch that video sometimes, just like, aw, that was a good story.
Hi, everyone. My name is Susan. My pronouns are also she, her, and I love top 10, usually because I get to talk about so many different types of science in one 20 to 35-minute period. But my other favorite part, hopefully you'll get to hear a little bit of it today, is when Eric comes up with the really clever puns to introduce each story. So pay attention to those.
Well, we had some better ones earlier in the countdown, but our first entry today could easily have folded its way to the number 1 spot in any year but 2020. Megan, do you want to tell us about our pick for number 2 this year?
Yes, I do, Eric. And that was so funny that I forgot to unmute myself. Yeah, so our number 2 story is about something called AlphaFold. And so AlphaFold is an artificial intelligence program that can figure out how what proteins look like, little molecules called proteins. And so this is super, super, super important because proteins are one of the most abundant molecules in living things. And they have all sorts of different functions, right? They're in our cellular structure, they're in hormones, enzymes, muscles, our blood, all sorts of things. And so I'm just gonna put up some slides here real quick. And so proteins are made of 20 different types of chemicals called amino acids. But unlike the string of beads that you see in this model here, proteins don't actually exist in nice straight strands. The chemical interactions between these amino acids, well, they cause proteins to fold into these really complicated, twisted structures. And the ability of a protein to function or to do its job depends on this twisty, folded 3D shape. So understanding how proteins fold is really important for understanding things like certain types of diseases and how they work. Now, the problem is that proteins are really, 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 really small. We can't just easily look at them with a microscope. And so it's hard to predict how a protein will fold or twist. And this is also because there are so many different types of possibilities or the ways that a protein can fold. And there are some laboratory techniques that can be used to solve a protein structure, figure out what it looks like, but these can be really, really time consuming and they can be expensive. So far, scientists have only been able to solve the structures of a really small fraction, so less than 1% of the 200 million known proteins. And this persistent protein puzzle is called the protein folding problem. And this is where AlphaFold comes in. So AlphaFold is an artificial intelligence program that was developed by a company called DeepMind, and it tries to figure out what proteins look like based on the sequence of amino acids that make up the protein. And in order to get this program to work, these computer scientists trained AlphaFold using over 100,000 examples of known proteins. And then AlphaFold could learn by example to figure out what unknown proteins look like. Now, after AlphaFold determines a protein structure, it can generate a computer model like this one of the protein's twisty, foldy shape. And to see if it's working, scientists test their models by matching the structures that AlphaFold predicts to structures that have already been solved by more traditional experimental methods. Now, in this image, the blue is the AlphaFold prediction and the green is the experimentally determined structure, and they match up almost exactly. But now to really test the power of AlphaFold, this team entered AlphaFold in a competition that's called the Critical Assessment of Protein Structure Prediction, or CASP for short. Now in CASP, programs like AlphaFold compete to see whose program can come the closest to predicting the real structure of proteins. And the competitors are judged by something called a global distance test. Now, if the structure the computer program predicts gets a score of 90 or above on this test, then it's considered a match. And AlphaFold blew the competition away at this year's CASP. Lots of the structures that it predicted scored 90 or above. And so this was a really huge breakthrough in the effort to solve the protein folding problem. And AlphaFold is definitely not perfect yet. It struggled with some really complex protein structures when they tested it out. But regardless, scientists are really super excited to put AlphaFold to work to help us better understand certain diseases and how to design medicines and vaccines and just generally better understand nature because proteins are everywhere. So it's just a really, really super huge breakthrough.
Wowza. Yeah, this one is, I mean, it takes a little bit of understanding, but this was one that when it came out, everyone I know in the biology field was just mind blown. I mean, this doesn't really happen every day. I mean, the complexity is so wild. What can understanding proteins better let us do? What's the like end of this,
I think there's a lot of things. I think the possibilities are a little bit endless. Some of the big ones are just better understanding how diseases work. And even if you look at something like SARS-CoV-2 that causes COVID-19, right? But lots of proteins in there, especially that like spike protein on the outside. Those kinds of proteins can be really difficult to figure out what they look like. And because this function of a protein is linked to its structure, being able to figure out what proteins look like is super important in terms of figuring out how to fight or treat diseases or figuring out how to design medicines and things like that. So those are some of the really big things that we can do. And right now, we've only been able to determine the structure of about 170,000 proteins out of the 200 million proteins that scientists know of. So this is going to probably help us figure out way more than we've been able to do so far through those kind of slower experimental methods. It's really super exciting.
Megan, we got a question coming in. Could AlphaFold help investigate sickle cell disease? I'm not sure if you're...
So possibly, I don't know the answer to that certainly, but sickle cell disease does have to do with proteins. I know that for sure. So in sickle cell disease, and correct me if I'm wrong, somebody, but I believe it's a problem with hemoglobin, which is the protein in our blood that carries oxygen. And so in sickle cell disease, there's a little mutation, like a little change in the person's DNA that causes them to have their hemoglobin to be a little bit different. And so it's the structure in the protein, it causes the cells to be in a sickle shape. And this leads to all of the symptoms that are associated with sickle cell and not being able to carry oxygen as well, and things like that. So perhaps being able to determine proteins like that, I don't know for sure if all of those proteins, I know things like hemoglobin were some of the earlier proteins that scientists figured out the structure of, but certainly perhaps programs like this can help us find out even more.
Yeah, I would say, yeah, as Megan said, hemoglobin was actually one of the very first proteins ever where we figured out what it looked like. So if we hadn't, AlphaFold would be 100%, would be so useful in knowing that. We happen to know a lot about sickle cell because it's a very well-studied disease. But think of all the diseases out there that we haven't as well studied as sickle cell. Absolutely. And that it's just a perfect example of understanding what a protein looks like can help us fix the disease.
I remember hearing something about this story that was that the sort of group that organized CASP, which is this competition that Megan mentioned, that's been going since 1994. I think they've been holding these competitions. And the group that first started it basically said this year, I mean, we're kind of done. There isn't really a need to do this anymore. I mean, this entire field of molecular biology is kind of, you can a little bit put it in the solved column, or at least closer to it, which is just not a thing that usually happens in science. It's this is an outlier for sure.
Yeah, people spend years on their PhD for trying to solve one protein.
Yeah.
And I'm sure they'll find other jobs out there, but it does put a bunch of these protein chemists out of a job. But then we can figure out, knowing the structure, what does the structure do? So maybe they're not out of a job.
Yeah, it's definitely not perfect yet. There's still some really complex proteins that it can't quite figure out, but it will certainly be incredibly impactful moving forward for sure. Yeah.
Wow. The stories covering it said that the AlphaFold was so accurate in predicting that sometimes they weren't sure if it was AlphaFold being slightly off or the actual experiments that measured them being slightly off. Those ones that are really expensive and take forever to get the exact structure. And that it's crazy that an artificial intelligence program can basically teach itself to do that. Can you talk a little bit about how that kind of program works, Megan?
Yeah, so the type of artificial intelligence behind AlphaFold is something called a neural network. And neural networks are really good at learning by example to recognize things like patterns but they have to be trained. And so the type of training that they used for AlphaFold was a whole bunch of known protein structures that they got from something called the protein database. So that has all of the known structures of proteins that exist. And so by showing AlphaFold these examples of known protein structures, it basically was able to start learning how proteins fold themselves. And now it's really good at figuring it out. And so it was really, really good last year. And I think one of the reasons that AlphaFold was so much better this year. And so the type of neural network that it is something called a deep learning network, which is like all these layers of neural networks, but they also added onto it something called an attention algorithm, or like an attention neural network is built into it that helps it sort of like build up things piece by piece too. And it's really kind of amazing how these artificial intelligence programs are able to do this.
I love the learn by example deep learning algorithms, because it would be like if a human could sit on the couch and watch baseball games for a year straight and then learn so much about how hitting is done that they just started for the Red Sox and only hit home runs all year.
Right.
That's basically how the computer program works.
Amazing.
Yeah.
So yeah, this was a really big story. It would have been #1 easily in any other year, and we'll be seeing one of those stories. We'll be seeing the impacts of it for years to come in the applications. But it's time to move on to the end of our countdown and our pick for the #1 science story of 2020. Not surprisingly, it's the story that impacted every part of all of our lives for most of the year, the COVID-19 pandemic. And there was so much amazing science that we wanted to discuss relating to the treatments that were developed, how fast and efficiently scientists were able to understand the virus and spread the word and even communicate with each other in a way that we've really never seen before across the entire world. But the thing we wanted to focus on the most as our number one story was the really amazing development of the COVID-19 vaccines, which are already rolling out, and we've seen millions of people vaccinated around the world. So Sarah, do you want to give us a kind of an overview of the timeline of those vaccines and the pandemic in general?
Yeah, absolutely. So this probably will not surprise anyone. This is our number one story, of course. This is huge. But I think that what's really great about this is to get a little bit of context for the vaccine, because usually vaccines take over a decade to create, and that's because there are many, many stages that come to sort of testing, but also because you have to sort of wait for funding, you have to wait for results, you have to wait for volunteers. There's a lot of sort of built into that. Now, the fastest that we've developed a vaccine was for mumps, which was a 4-year process. The picture here of that little baby is a baby receiving the first mumps vaccine, and that is the daughter of the doctor who developed it. So that was back in the late 60s, early 70s. And so for coronavirus, when this novel virus emerged, even we at the museum were saying, listen, summer 2021 at the absolute best. And that is still crazy fast. 18 months for a vaccine development is still wildly fast. But when we actually look at the timeline of what ended up happening, we see that things are way more exciting than we thought. First off, January 9th, 2020, that is the day that the World Health Organization confirmed to all the nations that are a part of it that there was a new coronavirus that is spreading from person to person. They named it SARS-CoV-2. 2 days later, Chinese scientists released the entire genome that had been sequenced of the virus, and they made it public. So that meant that everybody was able to access that genome. 2 days after that, the first vaccine was developed. Now, that might sound wrong because obviously we've only just started hearing about the vaccines, but it's because it is a special new type of vaccine that had been under development for a long time. And this is the first time that we're seeing successful use of that. We'll talk about that in just a moment. So that was 4 days after it was initially announced. Then in February 2020, phase 1 began of clinical trials. Phase 2 only took 3 more months. Phase 3 only took 2 months after that. By November of 2020, we were already hearing news that multiple vaccines have been rated over or near 95% efficacy. And then finally, in December 2020, healthcare workers around the world began receiving this vaccine. I mean, wowza. This is within 1 year. So January 9th, 2020 till December 2020, all within 1 year, the development of this vaccine, or rather these vaccines, took place. So, I mean, it's unlike anything that has ever happened in history. There has never been any sort of development this quickly of anything along these lines. So I know that there's a lot to talk about with how it happened so quickly. And there are some really, really great reasons behind it, but mostly it's just, I mean, nobody thought this. I didn't think this. None of you thought it. Don't tell me otherwise.
we were all doing the exact thing you said. Whenever anyone said, "Oh, we won't have a vaccine for a year," we all would say at least a year, much more like a year and a half, probably more than that. Putting on the
right, and even that is so fast. Even that is so much faster than anything we've done. Even a year would be wild. Even a year and a half would be wild compared to mumps. But here we are in 2021 with millions of doses already having been released. So
one of the most common questions we get is, how do the vaccines actually work? Because there's been a lot of news that they're like a new type of vaccine. So Susan, you want to tell us a little bit about exactly how they work and the different kinds of vaccines?
Sure. So I'm going to do a very quick overview of a vaccine, and then we'll talk about the ones that are out. So very briefly, to understand the vaccines, we're going to look at a virus. You can make vaccines against against proteins. You can make vaccines for bacterial infections. We're going to look at a virus. A virus is very simple. It's got genetic material, the like red curly stuff here, inside of a protein shell. And on the outside of that protein shell are more proteins that stick out. You've probably seen this before, especially with coronavirus. And that's what we're interested in, those green proteins here that are sticking out. So those green proteins that stick out, that's what triggers our immune system. And our immune system is what fights off diseases. So here's just kind of a typical immune cell, one example of an immune cell. And the virus, when it's inside of our body, it matches up with antibodies. If you've heard of antibodies, they're often either floating around or on the outside of an immune cell. They cause the immune cells to activate in the immune system to basically go into, hey, there's an infection, we're gonna clear it up and they make more of themselves. So a vaccine basically mimics, copies, or impersonates a virus without actually causing a disease. So very quickly, the types of vaccines that are out there, one of the most common is the live attenuated. This is the case where that little genetic material inside is changed, making it usually weaker so it doesn't cause disease. But again, those green proteins on the outside triggering the immune system. An inactivated or killed vaccine takes the virus and kills it, inactivates it, usually with heat or chemical poison, and breaks it apart. So it still goes into our body, green proteins still there, green proteins still trigger the immune system. Sometimes scientists, they figured out just a few years ago that they could make just the proteins and stick those into a person, and that can activate the immune system. A great example is the HPV vaccine, where they just use these proteins. Now, the Pfizer and Moderna vaccines are what's really exciting, where they take just a piece of the RNA from the inside and they package it into a little, a package. It's a nanoparticle package. This package carries the RNA into the body. RNA then gets changed into, RNA is its instructions, like DNA, RNA, it's instructions. Our body knows how to read instructions, takes these RNA instructions and makes the proteins, those green proteins once again. And then very similar is a viral vector vaccine. And this is what Johnson & Johnson and AstraZeneca are working on. And the viral vector, again, takes a piece of the RNA and puts it inside of a virus that doesn't bother us. So it doesn't cause disease. It's just basically a package again. So all those vaccines that are being developed for coronavirus, the reason they could be developed, 4 days after it was first announced, is because once you have the sequence, you take the sequence that you know is for these proteins and you stick them in a package. And these companies have been researching the package for years. This isn't anything new. They were just waiting for the RNA to put into the package. So that's why they were able to move so very quickly. And just if anybody wants more information on this, the museum is doing a lot of work on communicating more about the vaccines. So you can go to mos.org/coronavirus. And we'll be putting more and more information up there over the coming weeks and months.
So we've got 5 minutes left to talk about our number one story and how it just affected every part of our lives and the amazing science that came out of it. I guess I would say if you had only another minute or two to say one more thing about the vaccine or how effective it is, what would you want people to know, or what else are you just really kind of excited or impressed about?
I would say that the thing that I am most interested in people sort of knowing and understanding more widely is that the vaccines are super safe and they're super effective. We have gone through these tests and we've seen this. However, there are still a lot of questions remaining about how to distribute the vaccine. And that's not so much of the development of this vaccine happened. And again, vaccines, because as of now, there are nearly 70 patented vaccines that are underway being developed. But still, we're sort of, nothing like this has ever happened. We've never had to roll out a brand new vaccine across the entire world's population. And probably not surprisingly, we're not doing that good of a job of it right now. Not just in the United States, but all around the world. There are a lot of questions about who gets the supplies. How do we make sure that poorer countries that maybe didn't as much directly participate in the creation of the vaccine are still able to access the vaccine because they are still humans living on Earth. So there are still things that we're trying to figure out. I think that the discovery of the vaccine or the development of these vaccines is such a triumph of science. But now we're sort of sitting here with that question of what do we do with it? How do we take the good science we did and give it to everyone in the world is now the question that we're faced with. And I won't be surprised if that ends up being one of the big stories for 2021 is how did this go? What did it look like? And what did we sort of find out from it?
And I would say, and I kind of was already saying this, but I want to reiterate. So I've been looking forward to RNA vaccines being a thing for, yeah, you guys are nodding because you probably even heard me talk about it. Like,
for sure.
For like 4 or 5 years, at least now, have been just, it's going to totally revolutionize how we can vaccinate people. And I think, yes, it was very quick this year, but it's a great example that if a new novel disease pops up, or diseases we don't have vaccines for yet that we've known about for years, that it's being tested now, long-term effects and long-term use, but this is going to revolutionize the way we treat and prevent diseases for years. And so I'm just amazed and kind of excited that RNA vaccines are something that are going to be able to help so many people so quickly. It really is quite the number one science story, I would say.
Yeah.
And I think it might be the number one science story of the decade, or even
the decade.
I mean, century for a long time, until something bigger comes up. The impact that this vaccine development will have, and especially considering 100 years ago, we had a pandemic that just kind of had to run itself out because we hadn't even discovered what the influenza virus yet. We didn't discover what caused the influenza epidemic until years after. And in this case, being able to sequence the DNA in 2 days, come up with a vaccine, start distributing it the same year, it will be hard to top for another accomplishment of science coming up anytime soon. Well, that's all the time we have for today. Thanks so much for joining us all month long as we detailed our picks for the top 10 science stories of 2020. I'd like to thank my panelists and invite them to say goodbye.
Bye. Happy 2021.
And I'd like to thank all of you for tuning in and asking such amazing questions. We get lots of other digital content for you to enjoy, from our other live streams to our podcast Pulsar, which features an interview with someone who got their coronavirus vaccine and asking them what it was like, to short science videos about the technology of vaccines, town halls featuring experts. You can follow all of us at mos.org/mosathome. If you enjoyed today's presentation, please visit engage.mos.org/welcome to support MOS at Home. Thanks, everyone. Enjoy the rest of your day, and we'll see you next time.